well.
So it seems clear that with respect to a lot of the functions that you've been talking about for normal cells and for cancer cells, both of them are complex. Is it fair to say that the difference between a normal cell and a cancer cell is complex? In other words, is it a significant difference between the normal cell and the cancer cell, or is it a tiny change?
No, it's a sizable difference.
It's a sizable change, right.
And also, I think that this is in one property, in the enhanced growth, I expect to have similar large changes in genomic instability, for instance.
So the thing that confuses me is that it seems that complexity is something that develops in biology. The complexity is something, remember, I'm just a physicist, so I don't know anything about biology, but it seems that complexity in biology develops as a result of an evolutionary process. But the complexity is the result of evolution. That is, there's advantages to developing a particular complex system that are survival advantages.
Yes.
But cancer cells don't exist separately. So how did, that is, there aren't organisms that are separate cancer cells. It seems like it's something that happens that turns things on or off. So how does it turn out that a cancer cell gets this kind of complex difference?
This is speculation, of course, but what I think is that all the different pathways are pathways that have a normal use during embryogenesis. During embryogenesis, probably it's useful to use glutamine to make very quickly biomass, amino acid and then protein. The faulty thing is to reactivate this pathway in cancer. Of course, the mutation that reactivates in somatic cells this pathway is a mutation and therefore is out of the evolution.
Okay.
I think this is far from being proven, but it's very probable that we are making cancerous cell all the times, and we are making also some molecules which fight the cancer. So we are in continuous state of equilibrium. And what we know is much less than what we don't know. You mentioned the RAS mutation.
Yes.
The genetic RAS mutation in many diseases of cancer is very bad, and we know that it makes very difficult to fight such a disease. But nevertheless, and this is what I call singularity, there are many cases where for some patients, the RAS mutation doesn't do anything bad. Most problems are additional genetic mutation which fights this one. So what I repeat is that what we know is much less than what we don't know.
It would be better if you look at me, since I have to answer, because I cannot follow you if you look somebody else.
What I mean in a general way, that in biology and in cells, there are many more cases that something sounds peculiar because we don't know enough the various genetic aspects. That's all I wanted to say, because you mentioned the KRAS mutation, KRAS, which is generally considered very bad, and now in several laboratories, including our own, we also know the way how to fight those. So this is all the time, as you study more, you learn more. It's still not, I mean, systems biology will help a lot. But for the moment, for those of us who are trying partially successfully to fight cancer, in my case, it's now, we are just having in press, on the one hand, a way to treat pancreatic cancer, and on the other hand, to treat what's called the triple-negative breast cancer.
But,
because of fighting with more than one gun, fighting with 2 or 3 different things.
At the moment, two things. First, RAS. RAS is known to be activated in many tumors, from pancreas to others, and in many conditions, the survival of the patient is dependent on not having RAS activated. So RAS is a very bad oncogene in terms of survival. Second, presently the cure of cancer is on two lines. To use compounds that block reproduction of the cells, from adriamycin to cisplatinum, for instance, or to use the so-called intelligent drugs that are inactivating specific signaling pathway. Specific is the antibodies or the chemical compounds that inhibit the receptor for EGF. What I'm saying is that if it's true, and I think that is not only our work, there are a number of people working on the metabolism of cancer cells, and all are indicating that there is this very important rewiring. If the rewiring would be considered as a target for cancer drugs, probably is going to have a more effective response, given also the fact that the signaling molecules are very few in a cell. So to use a drug to inhibit few molecules in the cell is one thing, but the metabolic enzymes are present in much larger way, and therefore are a larger target. So it's like pointing to a small bersaglio or to a large one. So this is the reason why it would be useful to continue this kind of work. And of course, only when we would find out the complete picture for not only this property, but for other property, would systems biology show that it is very relevant for understanding complex phenomena. At the moment, nobody is able to do better.
My question was whether it is legitimate to talk about the cancer cell, or whether it's not like in schizophrenia that we have many, many different routes to the same final path. Is there something like the cancer cell, or are there many, many, many different variations that lead to the same uncontrolled growth?
The point of uncontrolled growth is that while you have the normal cells that when reach confluences, as said, or density, they stop one with the other, cancer cells continue to grow, and this is the reason the tumor is formed. This condition is dependent on metabolism. Unless you have a metabolism that allows the production of the building blocks, so amino acid, nucleotides, and so on, you are not going to have growth. It has been shown that if you take off glutamine, cancer cells in vitro die. But of course, you cannot take away glutamine in vivo, because glutamine is present in our bloodstream and is necessary for a lot of activities, for biosynthetic activities.
Thank you.
Thank you.
So